Today seems to be a vaccination day for India, 16th January 2021. And I thought of making a video to convey some of the things I know about vaccines and vaccine development in simple terms, and maybe answer some of the questions related to the benefits, the duration of the protection by vaccines, and other related questions such as the diversity of vaccine types or the efficacy of the vaccines. And this seems to be particularly bothering many people when only 10% or around that number of people are infected by the virus. How come we are looking at vaccines with only 70 to 90% efficacy to help us? You would hopefully find answers to these type of questions towards the end, and also find answers to questions related to immunity versus vaccines, etc. And maybe you would also agree with me that statements like this are very foolish. For example, vaccine making is a big business, and that's the reason why people have produced vaccine. This is impractical to think of as a possibility. And another foolish statement would be to focus on immunity so much that you don't take vaccinations, or you condemn it. You actually discourage others also from taking vaccines. That's totally wrong, and hopefully you will understand why I am saying so. Like any other living thing, our body is also made up of cells. We have lots of cells. One cell I have tried to show a cross section here. These are microscopic, very small, but viruses are much, much smaller. Viruses are non-living things. When a virus enters our body, it goes settle somewhere, meaning it enters one of the types of cells. Viruses can only enter some types of cells through recognition of certain proteins of the virus by our own cells, as I showed in one of the earlier videos. So initial interaction of the proteins on the outside of the virus, like these proteins, and our cell proteins is responsible for recognition. Anyway, we'll come back to that. We also have special cells in our body called immune cells, or let's call them soldier cells. They try to attack anything that is foreign always. Let me show the virus in a different way. Virus, when it enters our body, it starts dividing inside our cells. They divide inside a cell first, kill the cell, infect other cells, and again divide, and this continues. A lot of cells will die, body will be damaged. Meanwhile, our immune system multiplies the immune cells that target these viruses. But it is slow. By the time of course, one soldier cell or immune cell can actually target a lot more viruses, but they grow so fast, it's very difficult to control. And that's when we fall sick. 1% of infected people in India have died in COVID-19 cases. But if we understand the mechanism of this immune system, we can help ourselves, and that's the process of vaccination all about. So let's see how the viruses are recognized by our immune cells. They have to see them first, and obviously cells don't have eyes. There has to be some other mechanism, and that is by interaction, physical interaction. This immune cell here, virus soldier cell, part of it is shown. It has certain proteins which bind and recognize the virus protein, some of them. A similar interaction enabled the viruses to enter our cells in the first place, as I showed in the earlier video. But this is a little different interaction, which triggers the immune cells to be active, to produce antibodies, or to divide the immune cells themselves. That helps in interaction with virus cells in such a way, virus particles in such a way, that viruses are destroyed eventually. But of course, the interaction also helps the virus, our immune cells, to divide. But this interaction between the two proteins is critical. If we use this, if we understand this well, we could train our immune system before the virus gets in our body. That is called vaccination. So we could take the viruses out in a culture in laboratory and take out only the proteins. Now these proteins are not harmful. Proteins alone are not harmful to our body because proteins cannot divide. The entire virus particle can divide, not the proteins. So we take the proteins out, put it in a solution and inject. Inside our body, the proteins are recognized by our immune cells, and immune cells act by producing a lot of antibodies which will bind to the protein. There is also multiplication of corresponding specific immune cells which target only these proteins. Sometimes we have to give a second injection of the same protein, the vaccine. This is what a vaccine is in this case. So first injection is given, bodies are prepared. After some time, these cells start coming down because the proteins are destroyed in our body. Antibodies are also less in number. But again, you inject. For example, second time injection is given. Again, they work faster. Now body is trained better to interact with an incoming virus. But you did not inject the virus, you injected only the protein so far. So if a normal person who's vaccinated now sees the virus for the first time, the body is trained. So our immune cells or soldier cells are better prepared. So viruses start dividing, but our soldier cells divide much faster, produce antibodies, and counter all the viruses or virus infected cells within our body. All of them are killed and we don't feel sick. We don't even realize that there was a beginning of an infection. That's the effect of vaccination. The proteins alone, even from the virus, are not harmful. You can take any molecule separately from the virus, inject into our body, and the body is not hurt. Body uses these molecules to train itself. So we have to take out the proteins or other molecules and use them as vaccines. Let's see what other types of vaccines can be produced. So as I said, the proteins themselves could be used. We can produce them in the laboratory as well. If we know the structure of these proteins and other information about the proteins, we could use that information to produce them in the laboratory in large scale. And the proteins can be used in a solution as a vaccine. We can also make our body produce such proteins. I mean, our body cells are naturally producing virus proteins. When a virus comes in our cells, our machinery to produce our own proteins is used by the virus. I mean, nothing is thinking and doing these things. It's all happening automatically. For example, in our cells, we have a system where there is a biochemical code in the form of DNA. Some region in the DNA would be used to produce another molecule, RNA. It's like a message that is written in a biochemical form. That message is used by the cells to produce protein molecules that we need. Like hemoglobin is a protein produced in certain cells. Keratin is another protein that is used by our skin cells. Like this, all proteins required for our body are normally produced using the information in the DNA. When virus comes in, it uses its own RNA and its own genetic material like that, but our machinery to produce its proteins. Inside the virus also, we have an RNA molecule inside the coronavirus of SARS-CoV-2. So there are systems which can be exploited like this. Instead of protein, we could inject RNA or DNA, or some people use the dead viruses as vaccine material. In fact, that was one of the earliest ways of producing vaccines. When vaccines were discovered, the inactive forms of viruses like this were used by scientists. Edward Jenner in 1796 was the first one to develop something like vaccine for anthrax, smallpox. And Louis Pasteur, of course, is one of my favorite scientists. In 1881 and 1885, he produced two types of vaccines. And for a long time, people used the entire virus particle, which is, of course, dead. Now, that's what happens when we are infected, but we could inject in a systematic way the RNA molecule only from the virus into our system, and our systems produce viral proteins in a large quantity. If you want to know more about the types of vaccines, for a beginner, these references can be used. Of course, if you're a science student, you will have a way to find more important references. So please do find out more about that. Let's talk about efficacy now. Let's consider a vaccine of any type. The actual molecule, protein or something like that, is dissolved in a solution. Let's say XYZ solution. So this solution has to be injected into the body along with the protein or without it. So let's take one group, let's call them control group, where about 10,000 individuals are injected with only the solution, XYZ solution. And we study them. After about 1 month, we find among 10,000 people, 195 got the disease, COVID-19 disease they got. So you did not do anything. You just injected the solution and observed, and 195 got infected. So what's the ratio? 195 out of 10,000. So that is like per person, it is this number. Okay, just a ratio. So you divide 10,000, you divide 195 with 10,000, you get this number. So you got one number. Now let's go to the other group. Parallelly, we also did treatment. We gave the treatment to other 10,000 with the actual vaccine. So another 10,000 injected with the vaccine, which is in XYZ solution. And 10 of these people got COVID-19 disease after 1 month. So it's a general population, 10,000 treated with only the XYZ solution, another 10,000 treated with, injected with the vaccine, which is in XYZ solution. And here also you got a ratio now. So it is 10 in 10,000. So it is 0.001 ratio. Efficacy now I can calculate as shown here. I actually take the difference of the ratio in two numbers, two groups, that is 0.0195 and 0.001. I take the difference and divide it by the ratio in the control group, which is this. Let's do that. So the difference between this and this, as stated here, is 0.0186, and I divide this by the ratio observed in the control group, like this. Okay, so that eventually I can multiply by 100, I get 94 or 95%. This is the efficacy of this vaccine. This is how I calculate it. Now, when you have 70% efficacy, does it mean it is lower? It depends on where was the experiment done. If I might have done this experiment in Italy, where the overall death rate is higher, but if I do it in something like India or Africa or Sri Lanka, I don't know, any country where the death rate is lower, then in the control group beginning itself, the death rate is lower. So the variations, when you look at the efficacy, this is how I should understand it. If I have done an experiment in a country or a population where death rate is lower in the beginning, let's say I might get a number like this. Instead of 195 or among 10,000, I might get only 35 infected with 10,000. I mean, only 35 among the 10,000 infected. So the ratio would be different. In the control, in the actual test case also, it will be a little different, maybe something like this, or it may be 11, I do not know. So let's say it is 9 out of 10,000 infected for the test case with a different virus in a different population. Now, if I follow the same type of calculations, I will get a different set of number. It becomes 74.3%. But if this vaccine, which was tested in, let's say, a country in Africa, is tested this in USA or in Italy or in UK, I might get the same efficiency, a little less or a little more. We don't know. So it should be understood a little differently than simply as a percentage of people getting infected or protected. Okay, it's the, actually we can define it as proportionate reduction in the disease attack rate among the non-vaccinated versus the vaccinated groups under the phase 3 trial. So this is how it should be understood. If you, I mean, there are different ways of calculating it, but the original method was actually prescribed in 1915 by 2 scientists. The current efficacy that we are seeing, whatever 95%, 70%, etc., is based on short-term observations, because the studies have to be rushed, had to be rushed, and we have done this. Normally, nowadays, meaning in the recent years, whenever vaccination is being developed, companies would be scared to take up projects like that because it takes 10 to 15 years to do a project on that. To have tens and 40,000 number of people tested, and there is a lot of stringent procedure to be applied. And it costs a lot of money, and you have no guarantee. When I am having a company, for example, I have to test 40,000 people with all the rules and regulations following. In India, for example, we were slow, not because of some technical difficulty. We did not have enough volunteers who came forward to participate in the research. So it was very slow. So there are a lot of difficulties like this. So with 10 to 15 years of time for production of the virus, people wouldn't actually venture into developing vaccines that easily, and it's a risky business. But here, in this case, governments and companies and everything sort of came together, and they wanted to expedite the entire process. It's historical in a way in recent years that we were able to, most of the companies and the whole world was able to produce vaccines at such a high speed. But the downfall of that is they have not been tested for a long term. Okay, so we do not know for sure. It may be useful for later years also. Long-term immunization can be there, or only short-term immunity could be conferred by these vaccinations. Of course, the other thing to remember is 100% protection is impossible. Now, there are people who might claim that, question this rather, that vaccination is not 100%. So what is the point? That's ridiculous. But if you look at scientific data, there is no data available in any field that 100% protection can be guaranteed. But 100% eradication is possible. That's what I was trying to stress here. See, 1 in 100, let's say, gets infected. Now that person can start spreading the disease without his or her consciousness. That's the normal process. But even if you have 90% of the people immune because of vaccination, then the disease doesn't spread that much. And there are cases to substantiate this. So we have achieved success in case of polio, for example. If there is no other host around, the virus doesn't spread. Okay, so generally if how many people in the population get the disease and do not get the disease with or without vaccination, and there is data available for it. If you just want to find out, the data is there. You don't have to believe anybody's word, including mine. You just have to look up a little bit and then openly seek information rather than being biased. Okay, but eradication is possible by means of vaccination. Okay, and that's the hope we are having. And every reduction we can bring in is a big boon to society. You know, a lot of people have lost jobs or suffered because of by different means because of this COVID-19 spreading. And it's surprising a little bit how our psychology works, because in the initial days when there was no case at all, first case came in, there was such an excitement or anxiety and not excitement. There was anxiety and fear. Oh, there was a case in, there is a case reported in Delhi or Mumbai or Bengaluru. And now only 10 deaths in a state, or only 50 deaths in India today, one day, only 20 deaths. And we are relaxed. It's a little sad because the second wave, when it hits, again, same thing happens. That's what is happening in European countries. We don't know when the second wave hits. So people want to live normal lives and go back to normal lives. I'm not talking about wearing masks. That's the smallest thing we can achieve, we can bear. It's not a big difficulty or a punishment. I think wearing mask, keeping distance, we have to follow even after vaccination. For reasons, it's obvious, right? It's not 100% efficient. So you have to follow that. But that in combination with vaccination, we can actually have the normal business activities, normal educational activities, normal other society activities. And you should ask the the health professionals who have to wear the masks and then PPE kits even today when they go to hospital, and then they are scared to go back to their home, not thinking that they would be transmitting to somebody else. Now they will be relaxed because there is vaccination. And if we follow all the procedures after vaccination, there is no question of new wave, even though, of course, there is a surprise element that is always there. A new variant might come up anytime, but that possibility also is less if we take up vaccinations seriously. Okay, so that's the whole thing about vaccination. So we should all try to promote vaccination drive that is happening around. And we talk about strong immunity and all. That's not easily understood. In one of the last videos, I talked about 5 specific reasons why people respond differently, and immunity and other aspects were there, and there are a combination of those 5 also. So I think I talked about some of those in the last video. You might remember, so I was talking about specific aspects like this, and a combination of these also is critical in deciding whether a person is more susceptible or less susceptible and all that. So just to say that immunity booster is itself enough and vaccination is not required is foolish. I think we should really take vaccinations seriously and try to follow everything that is prescribed by well-studied principles. And that is the right way of going it. Let's stop the virus spread by correct information about vaccination rather than being biased about something and just trying to boost our egos by following something that we vaguely believe in, etc. So please help the society by spreading the right information, and let's stop the wrong information spreading. Thank you.